Chapter 11, it's about the muscular tissue. This chapter specifically talks about the muscles, the different types, and starts explaining the muscles at the molecular level. So at some point, we skip in the order chapter 10 because chapter 10 talks about the muscles too, but at the macroscopic level. So if you wanted to understand the function of the muscles at the macroscopic level, we have to talk about first of muscular tissue. That's why we exchange the order of these chapters. Now, before we move into the actual material of the chapters, let me actually tell you the key points for this chapter. So the muscle tissue has different levels of order, and within the muscle tissue we have muscle fibers, we have perimysium, myofibrils, we have fascicles, we have endomysium and epimysium. So starting from the most superficial order into the most deeper tissue or order, we have first epimysium, then perimysium, then we have fascicles, then endomysium, muscle fibers, and then lastly the most internal part of the muscles will be the myofibrils. So again, we start talking about the key points, and one of the key points is to know the orders of the structure of the muscles, which from superficial to deep will be epimysium, then perimysium, then fascicles, then endomysium, then muscle fibers, and myofibrils, the deepest layer. Now the muscles, the second key point will be that in response to a stimulus known as action potentials that arrives at the transverse tubules, the sarcoplasmic reticulum in the muscles will release calcium. So this is the second key point. So in response to the action potentials that arrives along the transverse tubules, the sarcoplasmic reticulum will release calcium. Then third key point is that after that, the muscle fibers will run out of ATP and calcium will begin to leak from the sarcoplasmic reticulum into the sarcoplasm, and this will lead to a condition in the dead that is known as rigor mortis, or stiffness of the muscles. Then the next key point will be that the cytoplasm of the neuromuscular terminal have vesicles that are filled with a neurotransmitter that is called acetylcholine. So this is the fourth key point. So that the cytoplasm of the neuromuscular junction contains vesicles that are filled with acetylcholine. And then the next key point is that because of the skeletal muscle contractions demand larger amounts of ATP, skeletal muscles fibers are rich in mitochondria. And this is the fifth key point. So because the skeletal muscle fibers, the contractions demand large amounts of ATP, the skeletal muscle have large amounts of mitochondria, and they are rich in blood supply. The sixth key point is that muscles are affected by hormones, and thyroid hormone will increase the energy use of the muscles, and this will lead to increased temperature in muscles as well. So again, this key point, the sixth key point, is that an elevated levels of thyroid hormone in the body affects the muscles by increasing the formation of energy in the muscles and increasing the temperature of the muscles. And then the next key point, the seventh key point, is that the heat energy produced from muscle contractions is released by the integumentary system. Now the eighth key point is that the muscles in our body, they have both slow and fast fibers. And slow fibers are abundant in the muscles that are slow, while the fast fibers are abundant in short muscles that at some point they have fast movement. So another key point will be that the nervous system that controls the skeletal muscles is the somatic nervous system. That's the last key point. So the last key point is that the somatic nervous system controls the skeletal muscles. Okay, so for the muscle, the muscular tissue, we have three types of muscle. We have skeletal muscle, we have cardiac muscle, and smooth muscle. And it's important to understand the composition of this type of muscles, not only at the molecular level and at the tissue level, but also at the microscopic level, so we can know how they function. In general, all types of muscle, whether they are skeletal, if they are cardiac, or they are smooth muscle, they have the property of being excitable cells or excitability because they can respond to stimulus in the environment. Some of these stimulus can be chemical, like receiving a neurotransmitter, or receiving a mechanical stimulus like stretch. And when these cells, the muscle cells, they receive these chemicals or this mechanical distortion or stretch, they respond by producing an electrical signal that moves across the plasma membrane. Another property of the muscle cells is that they have conductivity. So this electrical activity that or change that they generate after being stimulated doesn't stay within a confined area. These will be propagated along the entire muscle fiber, setting off waves of excitation. And upon conducting this electrical activity along the muscle fibers, these muscles will have the property of contractility or shortening in length when they are stimulated. Another property of muscle is that they have extensibility, so they are capable of being stretched between contractions. And they are elastic because this is a property that allows them to return to the original rest length after they have been stretched or contracted as well. So the first type of skeletal type muscle that we're going to talk is skeletal muscle. So a skeletal muscle is made out of cells, individual cells that are called fibers, and these skeletal muscle fibers are stimulated under voluntary control with your somatic nervous system, which is one of the branches of the nervous system that we have, and that it is one of the key points. So somatic nervous system stimulate the skeletal muscle cells or fibers so that they can contract. And these skeletal muscle fibers, they have a pattern or bands of striations, and this pattern can be seen in the microscope. Now, most of these skeletal muscles are attached to the bones. Some of them, like the facial muscles, the muscles of facial expression, they're attaching to the dermis, while another one, the actual diaphragm, the muscle that separates the abdominal cavity from the thoracic cavity, it is attached into the inner membrane within the division of the thoracic and abdominal cavities. And actually, out of all the skeletal muscles, by the way, there is an exception for the voluntary control. The diaphragm cannot be controlled voluntarily. So then this pattern of striations that we have in the skeletal muscle that is seen under the microscope, it is made by proteins that creates alternating light bands and dark bands. And these bands, they slide over each other, and sometimes they can create this overlap even without sliding to each other, that has these patterns that creates a darker place within the darker bands. So when we talk about control of the muscle, voluntary control, that means that you think about a movement and then you are capable of performing it. And the size of the skeletal muscle fibers in terms of length can vary. The average of the skeletal muscle fiber length is 3 centimeters, a little bit over 1 inch, but the fibers can be as long as 30 centimeters, like the ones that we have in the muscles in our leg. So this is a microscopic view of the skeletal muscle fibers. So each of these fibers that you can see here are individual cells. And as I said before, these skeletal muscle fibers, they have these alternating bands that create a pattern, so that these bands runs along the short axis of the fiber, which is this. And these dark bands are called the A bands, while these light bands are called the I bands. And these individual muscle fibers are separated from the rest by a membrane that is called endomysium. And these fibers, they are very long, they are cylindrical, and they have multiple nuclei, so they are multinucleated. So these are the connective tissue wrappings of the skeletal muscles that helps for protection. So we have a membrane that individually wraps each muscle fiber or muscle cell, that is known as endomysium. Remember, endo means inside, and actually mysium means muscle. And then several muscle fibers, when they are nearby, they form a collection of muscle fibers that we call muscle fascicles. These muscle fascicles are wrapped and protected by a membrane that is called perimysium. Peri means around, mysium muscle. And then several fascicles, they make an entire muscle. And an entire muscle is wrapped in a connective tissue membrane that is called the epimysium. Epi means above, mysium muscle. So it's the membrane above the entire muscle. And another connective tissue that it is associated with the skeletal muscles will be the tendons. The tendons helps to attach the muscles into the matrix of the bone. And these tendons are made of dense connective tissue, so dense collagen fibers that actually merge with first the periosteum of the muscles, and then they penetrate into the bone matrix. And this penetration of the tendons into the bones make this attachment of the muscles very strong into the bone, so that when you exert force with the bone, you can move the bone. And then the collagen fibers that are located within the skeletal muscle are somewhat extensible and elastic, that helps for the muscle to stretch slightly under tension, so it won't be so stiff, and also the muscle can relax or recoil, and then this will resist excessive stretching that prevents the muscles from injury, and it will help also the muscle to return to its resting length, and it will also help to contribute to the power of the muscle and the efficiency of the muscle. Now let's talk about the components of the muscle fibers. So the muscle fibers, they are separated from the environment by the by the plasma membrane. And the muscle fibers plasma membrane, it will have a specific name. The name is going to be called sarcolemma. So instead of calling the muscle fiber plasma membrane, you will call it sarcolemma. Sarco means flesh in Greek, and lemma it's for plasma membrane. Now, the cytoplasm of a muscle fiber is going to have also a specific name. It's going to be called sarcoplasm instead of cytoplasm. And inside the sarcoplasm, you will have proteins that are called myofibrils. And not only you will have proteins, but you will have also a source of energy for the muscles that is called glycogen. And glycogen, remember, it's basically these long branches of glucose that are going to provide the energy necessary for the muscles when you are doing exercise. Along with that, there is a special protein that is called inside the sarcoplasm that is called the myoglobin. Myoglobin, it's having iron, and this iron is going to give this reddish color to the muscle fiber, and this iron is going to bind oxygen, as many muscles require a lot of oxygen for performing their activity, and different muscle fibers, they have different amounts of myoglobin. So this is the components of the cells or the muscles at the different levels. So at the macroscopic level, this is a complete muscle, and this muscle is surrounded by this membrane that is called the epimysium. And this epimysium is continuous with this tendon that attaches the muscles into the bones. Now, this muscle is made by several fascicles. So each of these in here will be a fascicle. And fascicles are separated from each other or surrounded by this perimysium. This is a connective tissue layer. And then inside this fascicle, we have muscle fibers, individual muscle fibers, which is this one that it has been telescoped out. And then the muscle fibers, they are surrounded by the endomysium, and then inside the endomysium, you have the plasma membrane that is called sarcolemma. Now, the endomysium, as you can see, is not labeled in here, but it's not the same as the sarcolemma. The endomysium, it's a connective tissue layer that surrounds individual muscle fibers, while the sarcolemma is the proper plasma membrane of the muscle fiber. And this muscle fiber inside has these myofibrils, and these myofibrils, they're made of tiny proteins that are called the myofilaments that you can see here in this view. And then the sarcolemma that protects the muscle fiber also protects the sarcoplasmic reticulum, which is the calcium storage unit for the muscle fibers, and also it protects these thin and thick filaments, which are the myofilaments. Now, we're going to see more details about the sarcomere and the myofilaments, but the sarcomere itself, it is a unit or a segment that is located between two adjacent seed discs. And this sarcomere, it is what is considered the structural and functional unit of muscles. And then inside these sarcomeres, we have alternating bands of thick filaments and thin filaments, which you can see here. This is the ones in purple, these will be the thick filaments, and the ones in red will be the thin filaments. So the muscle fibers, as we said before, are the name of the individual cells in the muscles. And the skeletal muscle fibers, they have these multiple nuclei that are located inside the sarcolemma, pressed against the sarcolemma. And the muscle fibers, they are derived from stem cells that we call myoblasts. Myo is for muscle and blast for immature cell. Remember what I told you about this suffix of words. So whenever you hear the ending blast in cells, that means immature cells. So the myoblasts are the immature cells that are going to make the muscle fibers. And then during development, these individual myoblasts, they merge and they form a single muscle fiber, and that's why the muscle fibers, they end up with multiple nuclei. And some myoblasts, they don't fuse, and they form individual and specialized cells that are called satellite cells, that are going to be important for whenever the muscles get damaged, and then they help to repair the muscle fibers. Now, one characteristic of these skeletal muscle fibers is that they have abundant mitochondria, which are located in the spaces in between the myofibrils, and these mitochondria will be important to provide adenosine triphosphate for muscular contraction, or ATP. So what is the sarcoplasmic reticulum? It's the smooth endoplasmic reticulum that forms a network around each myofibril. And this sarcoplasmic reticulum is going to have a dilated end that is going to be called the terminal cisterns. And these terminal cisterns are going to run from one end of the sarcolemma into another in a cross section. And they will function to store calcium, and this calcium is going to be released every time that the muscle fiber is going to shorten or contract. Now, the sarcolemma itself, the plasma membrane, is going to send an extension deep into the sarcoplasm, into the cytoplasm. This extension of the sarcolemma is going to be tubular, so it's going to be an enfolding tubule that is going to penetrate through the cell and is going to emerge on the other side. And these T tubules, together with two terminal cisterns, one on each side of the T tubule, is going to form a unit that is going to be called a triad. Now, these T tubules are going to be important because these T tubules are going to help to transmit the electrical signal into the inside of the cell, the muscle fiber, so that these sarcoplasm terminal cisterns will receive also an electrical signal so that they can release the storage of calcium. So this is a better detailed view of the internal structure of an individual muscle fiber. So again, this wrapping in here, this will be the sarcolemma. This is the plasma membrane. And then here you have openings to the T tubules, transverse tubules. So the transverse tubules are going to run from this end of the muscle fiber, this edge, into the other edge. And this will be shown in here in this purple color, as you can see here. And then on the sides of these T tubules or transverse tubules, you will have these terminal cisterns, which are here in blue on the right and the left of this purple structure, which is the T tubule or transverse tubule. And then here, all of these extensions of the cisterns will be the sarcoplasmic reticulum. So when you have an electrical signal traveling into the sarcolemma, the plasma membrane, this electrical signal will deepen, going inside into the cells through these T tubules, and then the electrical signal is going to be transferred to the terminal cisterns so that the sarcoplasmic reticulum releases its calcium, because calcium is necessary for muscular contraction. And then in here you have the myofibrils with the alternating A bands and I bands, which are the striations that you see in the microscope. So the A bands stands for dark bands or anisotropic bands, and the I bands for light bands or isotropic bands. And then these myofibrils are made out of these myofilaments, which are proteins. So myofilaments are made out of myosin protein, which makes the thick filaments, and then actin protein, which makes the thin filaments. And each of these thick filaments made out of myosin have a shape like a golf club. So it has a shaft or tail that is made out of two chains that makes like a helix, and then it has a head. This head moves or flexes to connect to the thin filaments. And in the tip of the head, you have an enzyme that is called ATPase. And this ATPase is going to break ATP so that you will have this movement of the head after it has connected to the thin filaments. And then there is one part of these thick filaments where you don't have heads, and this is known as the bare zone. Now, the thin filaments are made of a protein that is called actin, and this actin is going to form also like a helix, so it's going to be two intertwined strands, and then it's going to have some units that are called globular actin or G-actin, and this globular actin will have an active site that is going to bind the head of the myosin, so that you have actually physical connection between the thin filaments and the thick filaments, forming like a bridge. Now, before contraction happens, you will have this long protein that is called tropomyosin that blocks the part where actin or the thin filaments binds to the thick filaments. And this tropomyosin is going to be connected to another protein that is called troponin. Troponin is going to bind calcium, and this calcium, when it binds to troponin, is going to make tropomyosin to move away from actin so that actin can bind into myosin. So I consider this similar to a chaperone. So troponin acts as a chaperone for tropomyosin. So when troponin is bound into tropomyosin, tropomyosin doesn't allow actin and myosin from binding each other. So I will show you later on in the lab portion how a video of how this connection and this regulation of connection take place between the thin and the thick filaments. Now, in the microscopic structure of the myofilaments, we also have these elastic filaments, which are made out of a protein that is known as titin. Titin is a springy protein that runs through the core of the thin filament. So in the actin, and these actin filaments are going to be anchored to the edge of the sarcomere into the seed disc in a line that we call the M line. And this will help to prevent the thin filament get stuck to the thick filament. So this elasticity, which is bringing back these thin and thick filaments into place, prevents the overstretching and provides recoil. So within the myofilaments, we have then the contractile proteins, which are myosin and actin, the ones that actually slide over each other. They make a physical connection, and then they disconnect and move back into their original place so that you can maintain the original length of the muscle. And then you also have these regulatory proteins like tropomyosin and troponin that at some point determines when the fibers can slide over each other and contract or not. And then in order for this sliding mechanism or this contraction between myosin and actin to happen, you have to have calcium. So calcium has to be released from the sarcoplasmic reticulum into the sarcoplasm so that the calcium can bind to troponin. Troponin moves away tropomyosin, and tropomyosin, when it's moving away from actin, these myosin and actin can slide over each other, and then they can form a bridge, and then you will have ATP available that can bind to the head of myosin. And then since you have these ATPase in the head of myosin, you can release the head of myosin over actin so you can have relaxation. So while calcium is necessary for contraction or shortening of our muscles, ATP is necessary for the relaxation of our muscles so that they can return to their original length, which was longer than before contraction. So this is how the structure of the different filaments look like. So this will be myosin, this one in purple. So individual myosin molecules are made out of this helix that you can see in the tail, and then it has this head that it is kind of a dilation and of the protein. And then in here, the region actually between the tail and the head is not labeled in here. It's called the hinge, because this head moves in this direction when it's contracting and then moves back into this angulated position when it's relaxed. So it tends to move the head towards the beginning of the tail in the hinge region, and then it relaxes, and then it stays in this angulated position that you can see here. And many of these individual myosin proteins make this thick filament. As you can see here, you have many of these myosins. And then here we have the thin filament. So this thin filament is made out of this G-actin, forming a filament. And then within this G-actin, you have these places that are going to allow the connection of the head of myosin into these groups. And then these groups are first occluded or obstructed by tropomyosin. And tropomyosin forms a complex with troponin. This protein, troponin, helps to bind calcium so that tropomyosin move away from G-actin, and G-actin can bind to myosin. And then when you have these two together, the thin filaments and the thick filaments, you end up with a structure like this, in which there is an area of this thick filament, the one in purple, that is called the bare zone. The bare zone doesn't have any heads, so you cannot have connection of actin in the bare zone, because the actual part of the thick filaments that connects to the actin will be the heads, the ones that you see here outside of the bare zone. And then this area where you have the thin and the thick filaments is the overlap, and this is the area of the muscle fiber where you have a thicker or denser part where you cannot see the light passing through the microscope. So this is why all this is called the dark band. So the striations or the bands in the muscle fibers results from the patterns or organization of the myosin and actin, and not only in skeletal muscle fibers but also in cardiac muscle fibers. And then the striations of alternating A bands, dark bands, with I bands will form this pattern. So what does A band stands for? For anisotropic, which is at some point the way that the light passes through these bands, and you can see them under the microscope. And then these A bands are surrounded in certain areas by the thick filaments, forming a hexagon. So you have 6 thin filaments surrounding a thick filament. And then in the H band of the A bands, you don't have these thin filaments. You only have thick filaments, so you have only myosin. And then in the middle of the H band, you have a line that is called the M line. Now the I bands or light bands stands for isotropic, which is the way that the bands reflect the polarized light. And then the C disc in the middle of the I bands will provide this anchorage for the thin filaments and the elastic filaments. And these will be located exactly in the middle of the I bands. So the sarcomeres are the functional and structural unit of the muscle, so it is the contractile unit of the muscle that runs between 2 adjacent C discs. And then when the muscle fibers contract or shorten, the sarcomeres, the C lines, become closer to each other, so the sarcomeres will shorten. And then neither thick nor thin filaments change their length during the shortening, only the amount of the overlap. So the thin and the thick filaments, they stay in the same length, but when they overlap, they stay in the same length, and then the bands, sometimes they look shorter. So this is an electron microscopy view of these bands. So here we have an A band with the H band in the middle, where we have the M line. So this will be the bare zone, the H band, and then these will be the light bands with the C disc in the middle, and then the area between 2 adjacent C discs will be the sarcomere, the contractile and individual or functional unit of the muscles. And this is the corresponding filaments for each band. So bare zone of thick filaments for the H band, in the middle of the A band, which is this, and in the A band you have a much denser area than the thin filaments, which is here, because you have thick and thin filaments overlapping. So that's why you see this dark band, because the light doesn't go through as much. And then these filaments in here are held by titin into the C disc, so they can contract and relax, and they can stretch back. And then this is the I band, which corresponds to this, and the C disc will be located in here. So this is what we have seen so far. We have seen the muscles from the macroscopic level out to the microscopic level. So this will be the myofilaments. In order for the muscles to shorten or contract so we can make a movement, we have to have a stimulus of this muscle. And this stimulus provided to the muscle is given by the nerves. So there is this connection. It's not a physical connection, it's a chemical connection between the muscle fibers and the nerves that is called a synapse. And this synapse, it has a gap that it is called the synaptic gap. So there is actually very closeness between a nerve and a muscle fiber, but there is a gap in between. And the muscle fibers will be stimulated when they receive a neurotransmitter, which is a chemical, from the nerve. So the nerves will release this chemical so that the muscle fibers can contract. In the case of the skeletal muscle fibers, they receive acetylcholine. This is the neurotransmitter released by the muscle fibers. And once this acetylcholine is released by the muscle fibers, this acetylcholine is going to travel through this gap and then it's going to bind into proteins in the skeletal muscle fiber sarcolemma, which are called the receptors, acetylcholine receptors, the nicotinic acetylcholine receptors. And then once this acetylcholine binds to this nicotinic acetylcholine receptors, these receptors are connected to channels that are sodium or potassium channels. And then once this chemical, the acetylcholine, this neurotransmitter, binds to these receptors in the sarcolemma of the muscle fiber, then you will open these potassium and sodium channels, and then potassium and sodium can enter into your muscle fiber so that the muscle fiber becomes less negative. And then it becomes depolarized. So the muscle fibers regularly are negatively charged inside. So they have a negative voltage. So when you receive this acetylcholine, then you open these sodium or potassium channels, and then this sodium or this potassium can rush into the cell, and the cell becomes less negative or more positive because sodium and potassium are cations, so they are positively charged ions, and then your cell depolarizes or becomes less negative. And this will make these muscle fibers to contract. And if you have a damage to the nerve cells, a physical injury or a chemical injury, the muscle becomes paralyzed. So you can end up with chronic damage to these nerve cells, which is denervation, and then your muscle fibers will shrink of size. So the muscle fibers will become atrophic. So it's similar as when someone has a broken bone. They immobilize this muscle, and then the nerves, they don't send as much signal so that the muscle can contract. So when you remove the cast from that broken bone, and then you see the size of the muscle in the limb of this person, the size of the muscle is going to be much thinner than the one in the left. Why? Because this stimulus of the nerve makes this muscle fiber to be at a certain thickness or size. And then this is not chronic, this is just temporary. So that's why when the person, after a certain period of time, after the cast has been removed, it regains its normal size. And different or individual muscle fibers can be connected to individual neurons. And the neurons that connects to the muscle fibers are known as motor neurons. So the motor neurons are part of the somatic nervous system. So you would call those nerves that connects to the skeletal muscle fibers somatic motor neurons. And these will be nerve cells whose cells bodies are located within the brainstem and the spinal cord that serves skeletal muscles. And then the somatic motor fibers and their axons lead to the skeletal muscle. And each nerve fiber branch out to a number of muscle fibers. And each muscle fiber will be supplied by only one motor neuron. And when one fiber connects to all the muscle fibers that it can connect, it will form a motor unit. So muscle fibers of one motor unit will be dispersed throughout the muscle, and they can contract synchronously in unison so that they can produce first a weak contraction over a wide area, and then they will provide the ability to sustain a long-term contraction as the motor units take its turns to contract. And then if you want to provide a huge amount of tension or force, you have to contract several motor units at once. So the average motor unit will contain 200 muscle fibers. And you will have some motor units that has less than 200 muscle fibers and some that they have more than 200 muscle fibers. Now, small motor units will be 3 to 6 muscle fibers per neuron. So each individual motor neuron will innervate 3 to 6 muscle fibers. And these neurons will have greater control over 3 to 6 muscle fibers. And these will be located in areas of the body where we produce fine control, like our hand muscles or the muscles of our eyes. And then large motor units will require less control, but they can have more strength. And then in this case, you can have, for instance, the muscle of our calf, which is called the gastrocnemius. You will have a single motor unit connecting to 1,000 muscle fibers. So in this case, you produce a gross movement. So this is how the motor unit looks like. So you have two individual motor neurons, like this one in purple and this one in green, and they connect to individual muscle fibers, and then they can produce this contraction of the muscle fibers. So the actual name or synapse between a muscle fiber and a nerve is called a synapse or neuromuscular junction. And the NMJ or neuromuscular junction, it has two endings. It has the axon terminal, which is the ending of the nerve fiber or the motor neuron, that has these synaptic vesicles with this acetylcholine, this neurotransmitter or ACH. This is one of the key points that I told you about, that the motor neuron has these synaptic vesicles filled with acetylcholine, the neurotransmitter, and then you have this gap or synaptic cleft that exists between the motor neuron and the muscle fiber. And then in the muscle fiber, the actual plasma membrane that has this receptor for acetylcholine is called the motor end plate. And each terminal of the nerve fiber within the neuromuscular junction will form a separate synapse within the muscle fiber. So one nerve fiber stimulates the muscle fiber at several points within a neuromuscular junction. So then what makes this motor neuron to release the synaptic at the motor terminal, what it makes to release these vesicles of acetylcholine? Well, it will be an electrical impulse that we know as the action potential. And then when this action potential reaches this ending of the motor neuron, which is known as the axon terminal, acetylcholine ACH is released into the synaptic cleft by exocytosis of the synaptic vesicles. And then acetylcholine will bind to the cation channel receptors in the motor end plate, which are the nicotinic acetylcholine receptors. And sodium ions will rush into the sarcoplasm. And then the cell will start accumulating positive charges, and then you will have a positive change inside the cell that is called depolarization, that is going to propagate from the surface of the sarcolemma into the T tubules, and then you will have this depolarization as well of the inside of the cell, and that is going to connect into the sarcoplasm to the terminal cisternae, in which you have release of this calcium, and the calcium will bind to troponin, and then troponin will make tropomyosin to move away from the actin, and then actin can bind into myosin, so you can have skeletal muscle fibers contracting by a process that we know as sliding filament mechanism. And then you have a cross bridge between the heads of the myosin and the actin G units, and then you will start the contraction. Now, muscle cells, they have millions of acetylcholine receptors proteins into the sarcolemma, and then when someone has less acetylcholine receptors, or they have these problems in the acetylcholine receptors, the person won't be able to hold the acetylcholine into these receptors, and then the person will have weak contraction of the muscles, like what it happens in a disease that is called myasthenia gravis. Into actually what happens is that the immune system in our body attacks these acetylcholine receptors. Now, within the synaptic cleft, we will have an enzyme that is called acetylcholinesterase, or ACHE, that will break down excess of acetylcholine that it was released by the synaptic terminal, and then this acetylcholinesterase is going to remove the excess of acetylcholine, and then this will allow also for the relaxation of the muscle, so that you don't have an over stimulus of the muscle fiber. So this is then the components of the neuromuscular junction. So you have these synaptic terminals here of this motor neuron, and then this will be the synaptic cleft, and this area in here of the sarcolemma will be the motor end plate. So if you magnify this, you will see this axon terminal, which is a dilation of the ending of the nerve, the motor neuron. And then inside this axon terminal, you have these little dots in green, which are the acetylcholine. And then you have this synaptic gap. And then you have this motor end plate, which is part of the muscle, which is this area that we have amplified in here, or magnified. And then you have these purple color receptors, which will be the acetylcholine receptors, that are chemically gated channels of sodium and potassium. So when this chemical, this neurotransmitter acetylcholine, is released by exocytosis by this axon terminal, and then acetylcholine binds into these receptors, these channels will open. They are regularly closed, and then they will open upon binding of the acetylcholine, and then they can allow the sodium and potassium to rush into the cell to depolarize the cell, so that you can slide these actin filaments over the myosin. And this is how it looks under the microscope. So these will be the muscle fibers, as you can see, because of the striations, and these will be the axon terminals of this neuron, motor neuron. So both muscle fibers and neurons are electrically excitable. So that means that they work with changes in voltage. And then electrophysiology will be the branch of medicine that helps to understand the electrical activity of cells. And then a voltage will be what we call an electrical potential, or difference of electrical charge from one point into another. And the actual resting membrane potential, or voltage, inside the muscle cells when they are relaxed, when they are resting, is minus 90 millivolts. So that will be the charge that these muscle fibers will have, minus 90. So they are negatively charged. And this charge is maintained by a pump that we call the sodium-potassium ATPase pump. So in an unstimulated muscle fiber, in a resting cell, there are more negatively charged particles inside. There are more anions than the outside. So that's why you have this minus 90 millivolts. And these anions makes the inside of the plasma membrane negatively charged by comparison to the outer surface. And then the plasma membrane will be electrically polarized with a negative resting membrane potential, or RPM. So the actual membrane potential of the plasma membrane is called the RPM. And then the excess of sodium ions of the extracellular fluid and the excess of potassium in the intracellular fluid will help to maintain this resting membrane potential when you have a functioning sodium-potassium ATPase. So what do you need to make this sliding mechanism of the fibers to happen? You have to have a change in this resting membrane potential. So you have to activate these muscle fibers. How do you activate it? By opening the sodium gates that are linked to these receptors of acetylcholine, so that you can allow the flow of sodium into the cells by going from a positively charged outside into a negatively charged inside. So since the muscle cells are negatively charged inside, the sodium will be attracted because the sodium is positively charged. Remember the law of opposite attracts. The positive will be attracted by the negative. And then these cations will override the negative charges in the intracellular fluid. So you will have the second portion of the action potential, which is called depolarization, in which the plasma membrane, instead of being negatively charged, become positively charged. And then after that, after you depolarize the cell, after the cell becomes less negative, the sodium gates close and the potassium gates will still be open. And then the potassium will rush out of the cell, partially repelled from the previous positively charged charges coming from the sodium, and partially because this potassium is so concentrated inside. So since the potassium is more concentrated inside. So since the potassium is highly concentrated inside and less concentrated outside, the sodium will escape. And then the loss of positively charged potassium ions will turn the membrane negative again. So you will repolarize the cell. And this will quick move up and down the voltage. So since your cell in rest was negatively charged and then becomes positively charged, and then through the escape of this potassium, the cells will repolarize or become negatively charged again. So how can you see the resting membrane potential? So it's seen as the waiting of an excitable cell to be stimulated. And the action potential will perpetuate itself from the area where it initiates along the length of the entire cell. So an action potential at one point causes another one immediately in front of it. So it will propagate. And this wave of excitation is called an impulse. Now, sometimes some chemicals can interfere with the actual propagation or actually generation of the action potentials. And some of these chemicals are toxins that can interfere with the synaptic function. So it can paralyze muscles. So we have some pesticides that inhibits the cholinesterase, the enzyme that prevents the degrading of the ACH, and that can cause what is called the spastic paralysis, in which the muscle continually stay contracted because you will have prevention of the degradation of the ACH. Another cause of this paralysis is when you have an infection by bacteria is called Clostridium tetani, which is going to cause tetanus or lockjaw. In this case, this is a spastic paralysis caused because this bacteria releases a toxin that is known as tetanus toxin that is going to block a neurotransmitter that is going to be called glycine. glycine is located in the spinal cord, and this normally stops the motor neurons from producing unwanted muscular contractions. But since tetanus is going to block glycine, these unwanted muscular contractions will happen, so you have overstimulation of the muscles, and then you will have a spastic paralysis too. Now, you can have also flaccid paralysis, in which the muscles are going to have less tone than what normally have, so the muscles will be limp and they cannot contract. A toxin that can make this is called curare. This will compete with acetylcholine for receptor sites, but it will not stimulate the muscle, so it will be blocking the acetylcholine receptors, so acetylcholine cannot bind, and then you will have flaccid paralysis. This curare is found in some areas on South America, and it was used by South American natives to basically kill animals, or paralyze animals. So it's a poison that it was used in blowgun darts. Now, botulism, it's a food poisoning disease that is caused by a bacteria. It's called Clostridium botulinum. In this case, Clostridium botulinum will release a toxin that is going to block the release of acetylcholine by the axon terminals, and then since you don't have acetylcholine, you will have flaccid muscles, flaccid paralysis. Now, botulism or the toxin produced by this Clostridium botulinum, it is used by many people, like in injections like Botox, to prevent the formation of wrinkles. So what are the four phases of contraction and relaxation? So you have an excitation in which you will generate an action potential in the nerve, and that is going to reach the muscle so that the muscle can produce an action potential. And then once the muscle receives this action potential, it's going to have this sliding of actin over myosin into what we call excitation contraction coupling. And then you will have the contraction, and then you have relaxation. What is contraction? So is when the muscle fibers develop tension and they may be shortened. And relaxation is when the stimulation ends and the muscle fiber relaxes and returns to its resting length. So this is what happens then. You have this arrival in muscle excitation of the action potential or nerve signal in this nerve. And then that will lead to the release of acetylcholine through exocytosis by the axon terminal. And then these acetylcholine vesicles are going to be released through exocytosis. Acetylcholine is going to be released into this synaptic cleft, and then you will have binding of acetylcholine, this neurotransmitter ACH in green, into these acetylcholine receptors, which are linked into these sodium-potassium channels. And then you will have opening of these sodium-potassium channels, so you have incoming potassium, and then you have incoming sodium and extruding of potassium. So potassium here will be represented by these blue dots, while sodium by these orange dots. And then you have this opening of these voltage regulated ion gates, creating an action potential in the actual muscle fiber. And then in the muscle excitation coupling, you have the generation of this action potential along the sarcolemma, and this action potential is going to travel into these T tubules. And then in the next step, calcium is going to be released by this terminal cisternae, and then calcium is going to bind to troponin, these little structures in yellow, and then this troponin is going to help to move away tropomyosin from actin, in here in red color, and then the myosin head can now bind into the active sites of actin. So you can see how tropomyosin has moved away from the actin binding sites. And then in muscular contraction, the first thing that is going to happen is that when you will have this myosin is going to create the head a bridge with actin, and you will going to hydrolyze ATP after you have this cross bridge between the heads of actin over myosin, and then this ATP that it has bound into the head of myosin is going to be broken down into ADP and inorganic phosphate, and then you will release this head from the actin, the head of myosin of actin, and then you have relaxation of the muscle. So this is what happens. Then you will slide actin over myosin because you had the movement of the head of myosin that has bound, forming a cross bridge with actin at its actin binding site, and then this will pull towards the center of the A band or the M line, and this will shorten the sarcomeres. And when ATP binds again, this head will release from actin, the head of myosin will release from actin, and then you will have the rupture of this cross bridge or the breaking of the cross bridge, and then you have relaxation. So ATP is necessary for relaxation, while calcium is necessary for the actual contraction. So when you stop sending acetylcholine, and when acetylcholine is broken down by acetylcholinesterase, you don't have more acetylcholine bound into the receptors for acetylcholine in the sarcolemma, in the motor end plate. So you will have the cessation of nervous stimulation, and then you reabsorb back the calcium into the terminal cisternae of the sarcoplasmic reticulum, and then you will have loss of calcium ions from troponin, and then tropomyosin can move back, obstructing the sites where the head of myosin has bound into actin. So this is muscle relaxation. So we have different types of muscle fibers. We have slow twitch or slow oxidative, and we have fast twitch or fast glycolytic. The twitch duration or the contraction between the muscle fiber types is different. So slow twitch will last as long as 100 milliseconds, while the fast twitch, the contraction will last as 7.5 milliseconds. So that's why they're fast twitch, because they contract really rapidly. The motor unit size is smaller in slow twitch versus fast twitch, and the motor neurons in the slow twitch are smaller, more excitable, and they're larger, less excitable in the fast twitch. In terms of contraction, how strong they are. Slow twitch, the contractions are weaker, while in fast twitch they are stronger. And in terms of ATP synthesis, the slow twitch, they have many mitochondria, so they have good production of ATP through aerobic metabolism. And then the fast twitch, they have mitochondria, but not as many. So they have mostly anaerobic metabolism. So the metabolism without the use of oxygen. And then fast twitch is poor resistant to fatigue, while slow twitch is fatigue resistant. And ATP hydrolysis happens slow in slow twitch. That's why it doesn't fatigue as easily, while in the fast twitch fibers, glycolysis happens really fast, so it runs out of ATP, or the hydrolysis of ATP is poor, so that's why it's non-resistant to fatigue. And myoglobin in slow twitch is it's high, so the fibers look more reddish than in fast twitch. And the glycogen content in slow twitch is low, while it's abundant in fast twitch. And mitochondria again is very abundant in slow twitch versus fast twitch. Capillaries, they provide a lot of blood supply to the slow twitch versus the fast twitch. So what is the color of the fibers? Red, because it has in slow twitch, because they have a lot of myoglobin, while it's white in fast twitch. Areas of the body where we have slow twitch, we have in the soleus muscle, we have erector spinae, quadratus lumborum, many of the muscles that helps for posture. Now, fast twitch, we have it in areas of the body where we require fast movement, like in the biceps brachii, muscles of the eye, and gastrocnemius. But remember, many of our muscle fibers, they have a combination of slow, or many of our muscles, they have a combination of slow twitch and fast twitch, but fast twitch is located in muscles where you require fast movement, like in our eye muscles. Now, so there is a relationship between the length of the muscle fiber and the tension and the muscle tone. So the length-tension relationship in muscle fibers will be how much is the amount of tension or strength generated by the muscle in comparison to its length, or how stretched or shortened it was before it was stimulated. So if you have a very strong contraction of the muscle before it is stimulated, you will have a weak contraction. When does it happen? Well, sometimes you can have an abnormal stimulation because your somatic nervous system is not working properly. So in this case, you will have an spastic paralysis. So you will have basically a spastic muscle, and then this spastic muscle is not capable of having a strong contraction. So if you're nervous, you become paralyzed because you are producing an overreaction with your nervous system, and you will have a weak contraction. So in this case, you have your thick filaments that just butt against the C discs. And if you overstretch your muscle before it's stimulated, you will also have a weak contraction. And this will be because you have a minimal overlap between the thick and the thin filaments, because you have put them farther apart, and you will have a minimal cross bridge formation. So in order for us to have the optimal tension or strength, when you contract a muscle, you will have a nervous system that is maintaining a normal muscle tone through partial contraction to ensure that the resting muscles are near their length. And this is the graph to show you the length-tension relationship. So this is the relative tension when you stimulate your muscles. So you can go from 0 up to 1, up to 2, etc. So these are units. And then this will be the length of the sarcomere before the stimulation. So if you have an over-contracted muscle, like in this case, see that you don't generate as many tension, as many strength. But if you have an overly stretched muscle, you have put the C disc farther apart, and the myofilaments, the thin filaments, farther apart from the thick filaments, which are this one in pink. So when you try to contract your muscles, when you stimulate it, these will have a minimal contraction as well, and minimal force, as you can see here. Why? Because you have flaccid paralysis in some cases. But when you have the normal stimulus, in which you have kind of half of the length of these thin filaments, the ones in orange, overlapping already these thick filaments in purple, and then you stimulate the muscle, you can generate the maximum contraction or tension that you can see in one. So this is the perfect generation of force when you have the normal resting length in your muscles. Now, when we die, 3 to 4 hours after, we generate this stiffening of our muscles that is called the rigor mortis. This is another point. Okay, so rigor mortis will be the hardening of the muscles after we die, 3 to 4 hours after we die. And this can be used to calculate at some point if someone has died recently or not. So what happens during rigor mortis? So during rigor mortis, you cannot relax your muscles because you don't have more ATP available. So your heads of the myosin will get stuck into the actin binding site in actin, and then you cannot break this cross bridge. But as the time passes by and the skeletal muscle fibers starts to degenerate because of the death, this enzymatic reaction that happens normally after we die with all of our cells, these enzymatic reactions will cause the leak of the calcium from the sarcoplasmic reticulum. And then this calcium will bind to troponin, and then tropomyosin is going to move away from actin, and then you will have this cross bridge between actin and myosin. So the muscles will be contracted but cannot relax because you don't have ATP. But then as the time passes by, you have the leak of the enzymes from the lysosomes, and then you start breaking down these cells of all the body, including the muscle cells, and then you start breaking down these cross bridges between actin and myosin. But rigor mortis can peak about 12 hours after death, then it diminishes over the next 48 to 60 hours. But at some point, the lack of ATP is what causes rigor mortis. And this is temporary because, as I said, the decay of the muscle cells will prevent the permanent formation of these actin and myosin cross bridges. Now, something happens similar when we are doing exercise. When we run out of ATP because we're doing a lot of exercise, we run out of glucose, we run out of ATP, we end up with cramps in our muscles. But as soon as we take a drink or something, a food that is rich in glucose, we start generating more ATP, and then our muscles relax. So then we have this part of muscle physiology that is called threshold, latent period, and a twitch. So with myograms, we can study this period. So in threshold period, you have a minimum voltage required to generate an action potential in the muscle fiber so you can have a contraction. And then you will have a twitch after you generate this action potential, which is a quick cycle of contraction and relaxation when the stimulus has reached the threshold or higher. But there is this latent period in which you cannot contract the muscle. There is a very brief delay between the stimulus and the contraction because you will have a time required for excitation, contraction coupling, and tensing of the elastic components of the muscle to generate an internal tension. So you will have then after this the contraction phase in which the muscle generates an external tension that is defined as the force generated to overcome a load and cause movement. Now, the internal tension in itself is going to be the normal muscle tone that we have, that it is produced by the stimulus of the somatic nervous system. But when someone put in your muscles a weight or a load, by itself you have to generate an external tension so you can overcome that load, and then you can move, and then you increase the tone of your muscles. And after you have generated this contraction phase, you move into the relaxation phase, which is the time when the tension declines to the baseline. Why? Because the calcium goes back into the sarcoplasmic reticulum, and then the myosin releases actin, and then the tension decreases, and this relaxation phase actually takes longer than contraction. But the entire twitch varies between 7 and 100 milliseconds. So then this is the time of stimulation. This will be the threshold. So you will have this latent period, and then when you reach this threshold, you have this contraction phase, which is shorter than the relaxation phase, as you can see here in the x-axis, which is representing time, and in here you have muscle tension. So muscle tension is generated at the peak of the contraction phase. And then that muscle tension, of course, gradually reduces during the relaxation phase. So it takes longer for muscles to relax than to contract. And then we have two types of contraction. We have one that is called isometric and another one that is called isotonic. So in isometric contraction, your muscle has a normal internal tension, but the external resistance causes the muscle to stay in the same length. And this isometric contraction will be a prelude to a movement when the tension is absorbed by the elastic component of the muscle. So you don't have this sliding of actin over myosin, but titin and other connective tissue can absorb this tension, and this prevents the muscle from changing its length. That's why it's called isometric. Iso means same, metric length. And this isometric contraction, what it does is maintain a normal length in our postural muscles, like our muscles in the back, and then this will help to antagonize the muscles that creates this instability in our body, so we can maintain joint stabilization. Now, in isotonic muscle contraction, the muscle will change its length, but no without changing tension. So you will have the same tone of the muscle, but the muscle will shorten. And we have two types of isotonic muscle contraction. We have one that is called concentric, that is when the muscle shortens, it maintains tension, like when someone is lifting weights. Or we can have eccentric, in which the muscle lengthens as it maintains tension. For instance, when someone is slowly lowering the length, lowering the weight. So at the beginning of the contraction, we will have an isometric phase. So you will generate muscle tension, but the muscle doesn't shorten. And then when the tension is overcoming the resistance of the load, the tension will level off. And then the muscle will begin to contract and move the load into the isotonic phase through the concentric and eccentric contraction. So this is the diagram to show you what happens. So when someone starts lifting its weight, the person might have the same length in the biceps, because this weight might not be sufficient to create an isotonic contraction. So this one will not generate as much tension. And in this case, the tension will be absorbed by the elastic tissue of the biceps. So the muscle will stay in the same length in isometric contraction. Muscle develops tension, but does not shorten. And then in isotonic concentric contraction, what it happens is, as you are lifting the weight, your muscle will shorten and the tension will remain constant, because there is not so much weight in this weight itself. And then when you are moving your weight in the opposite direction, when you are relaxing this muscle, your muscle will lengthen while maintaining the tension. And this will prevent the tearing of the muscle fibers. So what are the sources for ATP in the muscle? So the ATP supply will depend on the availability of oxygen produced by the mitochondria, and organic energy sources like glycogen, glucose, and fatty acids. And we have two main pathways for ATP synthesis. We have one that is called anaerobic and one that is called aerobic. Anaerobic happens within the cytoplasm of cells, and this will enable to produce ATP in absence of oxygen, and this will yield very little ATP and lactate, which is lactic acid. And this will be needs that will be disposed of by the liver. Now, in aerobic respiration, you produce far more ATP, and actually you generate it in the mitochondria together with oxygen, and this does not generate lactate. But this one, it's called aerobic because you have to have a constant supply of oxygen. So that's all for the skeletal muscle. Now, let's compare the different muscle types, because we not only have skeletal muscle. So we have cardiac muscle cells as well and smooth muscle cells. So skeletal muscle cells are mainly associated with the skeletal system, and cardiac muscle cells are only located within the heart, and smooth muscle cells are located in the walls of hollow viscerae like blood vessels, like the uterus, like ureters, etc. And then smooth muscle, we also have it in the iris of the eye, piloerector muscles in the skin. And the shape of the skeletal muscle fibers will be long cylinders, while the cardiac muscle fibers will be short, slightly branched cells, and the smooth muscle cells will be taper cells that looks like a spindle, so will be short fusiform cells. The cells that are usually the longest will be skeletal muscle cells, followed by smooth muscle cells, and then cardiac muscle cells. The widest cells will be skeletal muscle cells, then cardiac muscle cells, and the thinner will be the smooth muscle cells. Striations are present only in skeletal muscle fibers and cardiac muscle fibers, but they are not present in smooth muscle fibers. That's why they are called smooth. And in terms of the number of nuclei, the only muscle types with a lot of nuclei or multiple nuclei will be skeletal muscle fibers. Cardiac muscle fibers and smooth muscle fibers, they have only one nucleus, with the nucleus in the cardiac muscle cells near in the middle of the cell, and as well for the smooth muscle cells. So what are some of the properties that are shared by the cardiac and smooth muscle cells? So the cells are not as long and fibrous as the skeletal muscle fibers. They only have one nucleus, and the name of the cardiac muscle cells is cardiomyocytes. Both are involuntary, so that means that you cannot control them with your nervous system, and they receive innervation from the ANS, autonomic nervous system, not from the somatic nervous system. One property of the cardiac muscles is that they contract with regular rhythm, and they contract 24/7. And they are highly resistant to fatigue. And the muscles, they are contracting always in unison, so they contract synchronously. And the contraction may last longer enough to expel the blood. So they are stronger in terms of producing tension than the actual skeletal muscle. Characteristic of the cardiac muscle cells is that they also have striations, but the cardiomyocytes, the cells of the cardiac muscle, they are shorter and thicker, and the sarcoplasmic reticulum is less developed, but the T-tubules that they have are larger, and they can make also the passage of some extracellular fluid to go into the cardiac muscle cells. Now, the cardiac muscle cells are connected by strong joints that are called intercalated discs that appear as thick dark lines in stained tissue sections. And these cells, the cardiomyocytes, are electrically connected by gap junctions. So this will allow the muscle fibers of the cardiac muscle to have the same number of electrical charges. So that's why they can contract synchronously. And the mechanical junctions keep the cardiomyocytes from pulling apart. They are very strong connections. And One thing about the cardiac muscle cells is that once they're damaged, they don't regenerate. If you damage your skeletal muscle fibers, they can regenerate, but not for the cardiac muscle. And the cardiac muscle cell repair by fibrosis. So if someone has a myocardial infarction, the function of the muscle is not regenerated. And then the muscle fibers, they can contract without the stimulus of the nervous system, because they have inside some cells that are called the pacemaker cells that produces these rhythmic contractions that are generating this electrical activity that makes the muscle cells to contract. And these have this property, these muscle cells, the cardiac muscle cells, that is called autorhythmicity. So they're unable to contract rhythmically and independently. The cardiac muscle cells can increase their contractions or decrease their contractions by the action of the autonomic nervous system. And also they can contract stronger or less by the action of the autonomic nervous system, and they will produce very slow twitches. So they don't exhibit quick twitches like the skeletal muscle, and then maintain a tension for about 200 to 250 milliseconds. That gives the time for the heart to expel the blood. And the cardiac muscle cells, they uses only aerobic respiration, and they have cells that are rich in myoglobin and glycogen, and they have a very large number of mitochondria that makes up to 25% of the volume of the cardiac muscle cell, in comparison to the skeletal muscle cells that the mitochondria makes only 2%. So this makes this muscle to be, because of this, highly resistant to fatigue. So this is how the cardiac muscle cells look like. They are short and they are branched, and this will be the intercalated disc or the strong connections, and they only have one single nucleus per muscle fiber or per cell. Smooth muscle is named because it doesn't have striations, and then the muscle fibers, some of them, they lack a nerve supply, and others receive input from autonomic fibers or independent fibers with many varicosities that has synaptic vesicles with neurotransmitters. So this smooth muscle fiber is capable of repair its injury through mitosis and hyperplasia, so they can increase in cell number. This is the name for hyperplasia, or the meaning for hyperplasia. And then when you have an injury for a smooth muscle cell, they generate really well, they heal really well. And the smooth muscle fibers are slower than the skeletal and cardiac muscle fibers, so it takes longer to contract, but once they contract, they can remain contracted for a long period of time without fatigue. So since we have it in the uterus, females, and in reproductive age, these smooth muscle fibers in the uterus can contract, and they can contract longer without fatigue. That's how we end up with cramps during our menstrual period. Now, smooth muscle fibers, they can form layers within the walls of hollow organs, and this will help to contract and propel the contents inside the organs, for instance, the food within the gastrointestinal tract. And then as they contract, they can modify the pressure and the flow of blood in the circulatory system or the airways, so that you can have more entries of air or less. And then they can provide fine control in some locations, like in our iris, so we can control the size of our pupils, because you have contraction of the smooth muscle cells within the pupil. Or they can produce this erection of the hairs in our skin by the contraction of the piloerector muscles. So this is how these smooth muscle fibers look like. So these form a single unit of smooth muscle with this single autonomic nervous fiber that connects several of them, and at the tip of these muscle fibers, nerve fibers, you have these varicosities that are filled with these neurotransmitters that are released to stimulate these smooth muscle fibers, which, as you can see, they have a spindle shape. And where is their location? In the walls of hollow organs. So in this case, this is the cross section of the esophagus, the tube that brings the food from the stomach, as from the oral cavity from our mouth into the stomach. And if you see this cross section, these will be the layers of the smooth muscle fiber. So what is the name of the smooth muscle fiber cells? Myocytes. They have a fusiform or spindle shape. They have only one nucleus. They're located next to the middle of the cell, the nucleus, and they have thin and thick filaments, but they are not aligned or overlapping each other as the skeletal muscle fibers. And the sarcoplasmic reticulum is scanty, and there are no T-tubules. So what is needed for the smooth muscle contraction? Calcium that comes from the extracellular fluid by the way of calcium channels that there are in the sarcolemma. And then you don't have seed discs in these smooth muscle fibers, and they are replaced by something that's called dense body. And these fibers are well ordered, and you have these protein plaques on the inner face of the plasma membrane, and they are scattered within the sarcoplasm. So these dense bodies of neighboring cells are linked together, so forming a unit. So this is how the smooth muscle fibers look like. They are arranged in layers. So this is the longitudinal layer of smooth muscle, and it's here. So they are longitudinal, and then you have a transverse layer, which is this, and they can propel organs. So this is a high magnification, so you can see these long fibers with this central nucleus in the cells. And sometimes they form multi-unit, or they can form single units. And then this muscle, when they form a single unit, they have a gap junctions similar as to the gap junctions that we have in the cardiac muscle fibers. So this is all for this chapter. Feel free to email me any questions, and for some of you, have a good day or have a good night. I will see you in the next video. Bye-bye.